I blame physicists for this, for coming up with a unifying theory that's like trap everything. I don't know if we can do that about brain or not, because brain is true for physics as well, but brains were not like designed. They were evolved.
Once I got into dopamine more, I started liking it more because it has some idiosyncrasies that are very particular about dopamine. Sort of like the very simple story on the one hand, the very complex reality on the other hand, and the relationship between computation and experiment, and all of that makes this very enticing for me.
So this adds, I think, even more complexity that, like, what is the computational unit of the brain?
I actually think a little bit of the opposite, which is I think that AI and neuroscience are actually coming closer together.
This is Brain Inspired.
Hey everyone, it's Paul. Those voices you heard were from Ali Mohebi, who's at University of California at San Francisco, and Ben Engelhard, who right now is at Princeton but will be starting his own lab at the Technion Israel Institute of Technology. So Ali and Ben love dopamine, so much so that they are two of the organizers of the virtual dopamine conference that just had its second iteration. They also love each other, which unfortunately I had already stopped recording when they expressed that mutual admiration. A lot of love in this episode. But today's conversation was originally intended to be a precursor, sort of an introduction, for the panel discussion that I moderated at the recent dopamine conference. And that discussion that I moderated, you'll hear soon. It'll be the next episode that I release. But when people love something, they love to talk about it also. And so we talked. And it went longer than we thought it was going to go. And here we are with a full conversation about dopamine. So as you probably know, dopamine is a neuromodulator in the brain, and it's become kind of a darling in AI and in neuroscience because its release in the brain correlates well with a computational process called the reward prediction error and helps drive learning in that way. So during our conversation, you'll hear more about that. You'll actually get the backstory of how dopamine came to be important, how people have thought of it in the past, how it came to be the darling of reinforcement learning in AI, in its simple, elegant role, computing that reward prediction error, and how now, like everything else in the complex mess and or beauty of biology, it seems to be involved in plenty of other cognitive functions. So this conversation is a lot about dopamine and its various roles in cognition, and we talk a lot about those various cognitive functions. And we talk about how to think about the role of dopamine and other neuromodulators with respect to integrating them into a computational role with what we traditionally think of as computing events in the brain, like the spiking of neurons. And I also get their reflections on the deep reinforcement learning panel that you will hear soon. Thanks for listening and enjoy Ali and Ben. Ali, Ben, thanks for doing this. So, normally what I do is in the introduction to these things, I kind of introduce what the topic was about, but I thought it would be fun to have you guys on since you co-organize the dopamine conference and to kind of get your reflections on the conference as a whole, but also on this panel that people will be listening to in just a moment or two. So I guess, I don't usually do this either, but maybe can you just say a word about who you are and what you do? Ali, we'll start with you.
Oh, okay. So yeah, I can start. I'm Ali Mohebi. I'm a postdoc at UCSF Department of Neurology, and I am very interested in dopamine and what role it plays in motivation, learning, cognition, etc.
Okay, so I immediately want to ask a question, but Ben, maybe you could say just a word about yourself.
Yeah, so I'm Ben Engelhard. I'm also a postdoc, currently at Princeton in the Neuroscience department, and starting my own lab in a couple months in the Technion Institute in Israel.
Congratulations, man.
Thanks so much.
Although we were just talking a moment ago about how hard that move is going to be, especially in today's climate and just in general, selling cars, things of that nature.
Yeah, but with 3 kids, I'm sure it's going to be a breeze. So,
How did you guys get interested? So first of all, this was year 2 of the dopamine, the virtual dopamine conference.
Right.
Just give us the backstory of how the conference got started and tell us all about the super high demand that led to it being continued in year 2.
Yeah. So I'm gonna give a brief response and then let Ali take over, because the way I sense it started is one day Ali Mohebi woke up and said, we're in the middle of a lockdown. I think it was April 2020 or something like that. And he said, we can't do anything. We can't go outside. We can't have a conference. We can't meet people. Let's change that. And so he basically recruited a few people, either students or postdocs. We were 5 in total. And he said, let's make a conference. And so that's how we started. And then we ended up organizing everything pretty quickly. We started contacting all the speakers. We had an amazing response. We contacted, some of the best speakers in the field, and pretty much everyone said, yeah, I want to do it, because nobody could do anything, I guess. So, we were the only option in town. And that's how the first one started. And I think that because it was available to everybody anywhere in the world, we got over 2,000 people registering, which is a pretty large number for sort of a niche, neuroscience conference, for the subject of dopamine, not just, neuroscience in general, but just dopamine.
Yeah.
So that was a huge success. And also the quality of the talks was very impressive.
So Ali, did it come to you in a dream? So you just woke up and it was there, huh?
So I live in San Francisco, which means that I'm locked in a teeny tiny studio box. And then when we went into lockdown, I was going crazy. I'm just bouncing off the walls.
I thought you were going to talk about a psychedelic experience, because I thought you were going to go that route there.
But I think my friend Christine Liu, at some point, she suggested this idea on Twitter that, our dopamine meeting is going to get canceled. We were all supposed to go to Montreal for this dopamine meeting. And then she was like, it would be nice if we have a gathering of a few of us enthusiasts. And then during a psychedelic trip, perhaps, or something, it came to my mind that what if we do this conference? What if we put a conference together, a virtual dopamine conference? And I reached out to Christine, and I'm like, Christine, let's do this. It was an amazing experience for all of us.
Yeah. So the dopamine, so my role in this was I moderated this panel that people are going to hear that was based off the premise of the explosion of modern deep reinforcement learning, and kind of how that interacts with the rest of the dopamine story. Which is, I don't know how much of the conference is focused on the sort of systems neuroscience part of dopamine versus the reinforcement learning side. It seems like a pretty big blend of all of it. But how did you guys get into dopamine? Dopamine was something I always avoided when I was going through a computational neuroscience program, because it was too systemsy, too wet. But it was everywhere already back then, dopamine, but I was like, it's unimportant. So how did you guys get into it?
So I got into it by mistake. My PhD had nothing to do with dopamine. I was working on cortex, doing cortex-like things, brain-machine interfaces and learning and stuff like that. And I think the link to learning a little bit maybe led to it in that direction, but it was mostly when I was looking for postdocs in the US. And I traveled all around the US and talked to a bunch of people. And I started to think what I want for my postdoc. And what there was at Princeton was this sort of, for me, an amazing opportunity working with people like Ilana Witten and David Tank in a virtual reality system with mice and sort of cutting edge technologies. And it was about dopamine, which is related to learning, which is something that I would care about. So, all these things together, I said, let's do this. And once I got into the dopamine more, I started liking it more, because it has some, I would say, some idiosyncrasies that are very particular. Even within the system neuroscience field, there's something very particular about dopamine. Sort of the very simple story on the one hand, the very complex reality on the other hand, and the relationship between computation and experiment, and all of that makes it very enticing for me to study.
that the very simple story that you referred to there is what deep reinforcement learning is kind of based on. It's one of the most quote unquote successful stories in bringing together things like artificial intelligence and things like neuroscience. That dopamine underlies a reward prediction error that leads to reward, and there's very specific circuitry associated with it. But the other thing that you alluded to is that that's not the only story, and we talk about that some in the panel as well. But so I just wanted to make sure that I hammered that home. That I believe that's the particular story you were referring to.
Yes, it is. TD learning. The ideas by Schultz showing the reward prediction error response of dopamine neuron and how it relates to learning. Then the idea that this is basically a signal that's propagated, throughout the brain and mostly the striatum. The signal goes, let's say, to ventral striatum, the accumbens, which is sort of considered one of the sites for learning. And then you potentiate the corticostriatal connection, the synapses, based on this reward prediction error signal. And that's how you learn. And, that works great for simple things like Pavlovian conditioning. And there's been a bunch of experiments showing that. But the more we learn about the circuitry and the more we do behavior that is more complex, and the more we look at, the brains in general, we see how this simple story is starting to become stickier and stickier.
Ali, is that sad or is that a happy thing that because dopamine has a bigger role now, we think? Or is, okay.
So I want to have a revisionist history of dopamine.
Okay.
Nice.
I think we should go back to where it all started from, right? I mean, so if you look at dopamine and what did we know about dopamine and when? I mean, dopamine, we knew at some point that had something to do with movement, right? And we knew this, like many other things in neuroscience, by a malfunction. So we know that in Parkinsonian disease, patients, they lose their dopamine cells, majority of their dopamine cells, and something becomes wrong with their movements, right? So for a long time, people believed and were looking for movement-related signals in the dopamine. And then, at around the same time, there was this other idea of dopamine having some role in learning from rewards, which goes back to like Olds and Milner's stuff in McGill, where they would put they would stick an electrode in the rat's head, and once in a while, when rat is visiting one corner of a cage, they would zap him or her. And then they would see that rodent, the rat, is more interested to explore those corners of the cage. So, and then they realized that depending on where exactly they stick this probe, the likelihood of rodents visiting those corners becomes more and more. And when they put it in like midbrain VTA dopaminergic cells, I mean, this is where the rats start visiting that corner more and more often. So people started hypothesizing that dopamine has something to do with reward. So these two accounts start to like develop mostly like independently. One is more like in the psych departments, and then on the neurology side, people interested in Parkinson's disease started looking at dopamine and interpreting dopamine in that sense. And so, I mean, another thing that perhaps inspired by Skinner and Walden too was that there's some aspect of reinforcement in this dopamine signals, because like Olds and Milner were able to teach the rats to press a lever by linking a lever press to a zap in the dopaminergic system. So you stick an electrode, zap them, they learn to press a lever. So there's now some reinforcement that motivates you to go and push the lever further and further.
You referenced Walden too. Was it? I read that book many years ago before I was a neuroscientist. Is it actually in that book? And is that a good book? Should I reread it? I don't think I should, but
maybe you should reread it. Yeah, I read it like year or two ago, and it was interesting. I think if you have ideas about how to shape a society or how to like reinforce acts or values and virtues that you want, and is it even possible? I think it's a good idea to revisit that book.
But it's a novel, right?
It's a novel by Skinner, right? So
yeah,
he
B.F.
was
Skinner.
Right. So, I mean, this whole idea of how you can reinforce things and potentiate acts and things that you value more, right?
Mm.
So, and like here, it's like quite related also. I mean, these whole series of experiments, another reason they got a boost was that this is a good animal model for studying addiction. That you get some stimulation in the dopaminergic system and you keep going back and back to the sites that you received this stimulation, or keep doing the acts that led you to this feeling of it. So this is how like dopamine got involved in the addiction research and in the motivation. But then these fields evolved separately from each other. And Wolfram Schultz, actually, in his last year's talk at Vida, he started going through this very brief history of dopamine, that when he started looking at dopamine, he was after the movement signals. He had like monkeys sitting on a chair and moving these bars, doing center outreach tasks and such. And to his surprise, there were like absolutely zero dopamine cells that would respond to the movement itself. And they were confused. He was like, yeah, I'm recording from muscles. I'm recording from mouth. I'm recording from eyes. I see movements. I see movement signals in the EMG of these muscles, but I don't see anything in the dopamine cells. And years later, I mean, with the advent and popularity of reinforcement learning, people started looking at and reinterpreting Schultz data, and they realized that it might have something to do with the reward prediction error. So I think, the dopamine story did not start. So this is the revisionist part. It did not start with the prediction error.
No, I agree, but and I agree with what you said about sort of the diverging views about the movement, Parkinson studies, and sort of the addiction / reinforcement studies. But I think that at least at some point, and probably even now, but certainly, previously, people sort of thought about, because we know that there are different sort of canonical, pathways of dopamine. More typically, what's called mesocortical limbic, perhaps, which is, VTA to accumbens and then to cortex. And then perhaps the nigrostriatal, which is, substantia nigra, pars compacta to striatum, at least the dorsal part. And so I think that people did associate sort of the nigrostriatal pathway mostly with Parkinson, because in Parkinson, dopamine neurons who die are mostly in the SNC and not so much in the VTA. In fact, there is sort of like a line that sort of divides, and you can actually look at it if you stain for something called calbinding, and actually see the difference between SNC and VTA dopamine neurons. And the SNC one dies, the VTA do not, and the SNC have sort of some of the different projection patterns. So I think people associated the movement / Parkinson function with the depth of these, SNC, substantia nigra dopamine neurons, and perhaps even having a permissive role rather than a proactive role. So the idea is that maybe if you have enough dopamine in striatum, because these dopamine neurons are giving you this sort of tonic signal, right? So this constant release of dopamine, maybe that's enough to have movements, and that's all you need. And then maybe that's why you're not seeing any movement signals in these neurons, perhaps. And the VTA neurons, the sort of mesocortical limbic pathway, that's the one that has been more associated with reinforcement as well as addiction and perhaps motivation.
I don't disagree. I think one of the appeals of the dopamine story in the past was that how beautiful, there's like a unifying theory of dopamine cells as a global broadcast signal. They're just sitting there, and if you read some of Schultz's older stories, or even others, have been proposing dopamine as this broadcast signal, that these cells are just sitting there regardless of which type they are, recording from SNC cells in the case of Schultz. Most of actually his recordings were from A8 and SNC, and then some from VTA. Majority of them do send this broadcast reward prediction error signal, and I think that's one of the beauties of this theory. But then in recent years, there's people are paying more and more attention to different functions of dopamine, such as yourself in your 2019 paper, where you start talking about different roles that dopamine cells have in different functions. So I think it's exciting times to be studying dopamine, because we are going back to some of our original thoughts, Because like the appeal of this reward prediction error theory, I think, was so much, and it made us like focus a bit too much on one aspect of dopamine function for the past maybe 20 years. And like early on, another aspect of dopamine function that I forgot to mention is its role in cognition and cognitive aspects like working memory, attention, and through the work of one of the early works from like late 70s from Pat Goldman-Rakish and her colleagues at Yale University was that how dopamine in the prefrontal cortex plays a role in like sticking to ideas, keeping something in your mind, right? And like she and her colleagues ran a bunch of experiments like increasing dopamine in the cortex, specifically in the prefrontal cortex, or ablating dopamine in the prefrontal cortex, and see how like these like working memory function get affected, and they link it to some mental health disorders such as like ADHD, schizophrenia, and others. But this work is also, I think, has been overshadowed by this success of the dopamine equals reward prediction error theory, and people are slowly going back to those ideas, using new tools and testing some old theories, coming up with new hypotheses, etc.
Well, this is why I asked, though, if it's a sad thing, because there's an attraction to the simplistic explanations of, because it's a simple explanation is powerful, right? Dopamine is this, and of course, but the problem is it's never that way, at least in the brain. But I can't tell you how many times I've been reading. There's a story about two brain areas that are connected in some directional way, and then just a few years later, it's always actually they're also connected bidirectionally and through three different pathways, and it just always just explodes into the mass of complexity that is the problem, that is the challenge of facing us.
Yeah, I think that naturally, I would say we tend to look for this sort of simplified explanations because that makes it easier to understand. Otherwise, it's very hard to just from the very get-go look at something that's incredibly complex and make sense of it. And with regards to what Ali said about sort of the working memory by Goldman Rakic and others, that was also, together with the movement story, that was also, at least in part, I think by them too, associated with sort of the idea of a tonic dopamine signal, sort of like a very slowly changing dopamine release. And that was contrasted with the sort of phasic signal, which was like a very strong, rapid burst of dopamine neurons, which was associated with the reward prediction error. So even then, when the dichotomy was known, right, because it was known, as Ali said, from the 70s, I think 79 was the main paper that was published by Goldman Rakic. It was known that there is an effect in working memory and there is an effect on movement. People still say, you know what, there's sort of like these two dopamine signals, the tonic one that sort of like changes very slowly and has a mostly permissive role, sort of allowing the downstream circuits to do their thing, either movements in striatum or perhaps working memory in frontal cortex. And then you have this sort of phasic signal, sort of like rapidly changing, rapid bursts that would have a role for reinforcement. And again, of course, that cannot, it's not going to turn out to be the case, because as you said, things are always more complex than they seem, but I think it makes sense to start with some, so I'm not criticizing, the past, because it makes sense to start with that. You need to have a starting point where you can make sense of things. And it's not wrong to simplify as long as you keep an open mind that it's probably going to be more complex than your sort of simplified explanation.
This is a topic for another conversation elsewhere, but maybe I should just mention for the reference that I don't believe in tonic dopamine, right? But we can discuss it elsewhere.
Oh, you don't believe in it?
I don't believe in it. I wrote about it. Yeah.
You have to address that now. Yeah. It's a slight diversion, and that's fun. The antagonism is good for the podcast.
Let's diverge.
Yeah. Okay, let's diverge. I don't understand what tonic dopamine means. With the dopamine signal, what you measure is either firing of dopamine cells, right? Dopamine cells have a tonic firing. That is, and they're pacemaking cells. They fire like 3 or 4 spikes per second on average. Some of them a bit more, some of them a bit less. But then sometimes they fire these bursts of spikes. And that's how they do. You don't see dopamine cells changing their baseline firing rate at all. Like I've recorded from a bunch of dopamine cells. I haven't seen anything like this. Uchida's lab have recorded, Jeremiah Cohen and others in his lab have recorded from a bunch of dopamine cells. You don't see at any point in time dopamine cells change their baseline firing, right? So we don't see it in any context. We have tried it a few times. I mean, of course, absence of evidence is not evidence of absence, but we don't see it in any context.
I'm surprised by this argument coming from you, Ali, because you're one of the main people that are sort of exploring this idea about how actually the release of dopamine in the axons of these cells might not be related in some cases to the firing of the cells themselves.
Exactly.
So you can still have fluctuations based on sort of axonal processes like maybe acetylcholine, right, which I think you're explored as well, and other people too. And so you can have sort of a baseline 5 hertz or 4 hertz firing, which will give you a relatively constant with some noisy fluctuations of photonic dopamine release. And then you could have sort of modulation of that release, perhaps with some sort of axonal processes. And of course, we don't know how much modulation we actually need because, again, if it's a permissive role, then it's enough to have maybe the 5 hertz gives you what you need to have movements. And then maybe there's some modulation on top of that, which will make movements, change them a little bit. I think with Golan and Rakic, they had this inverted U-shape sort of idea where you have too much dopamine is not good and too little dopamine is not good, and sort of, the Goldilocks idea of dopamine for working memory. So maybe the 5 hertz is sitting somewhere, around the Goldilocks zone, and then you can have sort of axonal processes that would modulate that. So that's why I was surprised by this argument coming from you, of all people.
Okay, just to answer your surprise, and then maybe we can move on. I mean, this could be like a never-ending conversation, or I'm happy to chat further. But, I mean, yes, so that's the second part of my argument, that now when you look at dopamine release, you don't, again, see slow events. So with tonic, what people often mean is that there's this like inherently slow change in the signal. When we look at dopamine release, there's also, now that we have the tools to look at them like in a very fine temporal resolution, you would see these like rapid bursts of activity or like slow, very fast changing. So one thing that I think, and we think in our lab, that might have alluded to this tonic modulation of dopamine concept was that our measurement tools of dopamine release were slow. So if you measure a signal slowly, you may deduce that it's a slowly changing signals. But as you start using better and better tools with better temporal resolution that's made available just in the past like 2 or 3 years, we don't see slow changes in dopamine signal.
What is slow versus fast here? Because a spike is fast and a neurotransmitter is slow, but it depends on how.
That's exactly one of the. I think one of the reasons I think this tonic dopamine thing is ill-defined. That no one can give a good time scale of what is the time scale of a phasic versus tonic dopamine. If it's changing faster than or slower than 20 hertz, is it now called tonic? Or
tonic is a time scale of microdialysis, Ali.
Right, because in microdialysis, you measure like dopamine concentration every 10 minutes. So it could be, I mean, this whole notion could have been just a...
No, but obviously I'm joking, but look, for example, in cortex, we know that the reuptake of dopamine is pretty slow, right? Compared to, for example, the striatum, where the reuptake is pretty fast. So
or ion channels in a neuron membrane, right? That's
right. So you can well imagine that even if you have sort of fast signals, in dopamine release, because of the slow kinetics of the reuptake in cortex, for example, you could end up with sort of like smeared slower changes in dopamine concentrations. And the other point is that I don't think that the theory required, for example, by Goldman Rakic, I don't think the theory is required, like very precise modulation of the tonic signal. The idea is that, so you're in a bath of dopamine, and, if you have a good enough level, within a range, you can do, your working memory is gonna work fine, right? So you were talking about the modulation of the tonic signal, But I don't think that was ever really a strong focus of either of these theories.
No, I mean, yeah, that goes into, I mean, many of those studies, I mean, these were like super illuminative and like insightful for me, but they were using like late 70s tools, which mostly is like slow pharmacology. I mean, dump bunch of dopamine, in, some part of the brain, let it stick around for a few hours and see what effect it has on like activity of these cells, which is very different from its physiological time course. And now I think with better tools like optogenetics and other things, we can start like dissecting dopaminergic signals in the frontal cortex within the physiological time range that dopamine is affecting it. And in fact, that's something I will be doing in the next few years.
Oh, very nice. That's go ahead.
Yeah, no, I just want to say again a little bit, playing grumpy advocate, that it's true that, optogenetics in terms of the temporal resolution is so much better than pharmacology, but in terms of the, I don't know, like cellular resolution or maybe spatial resolution, you're still, when you're doing, for example, activation, you're activating in a very non-physiological way, right? You're activating the entire bundle, super strong. So I think that we don't really yet have, if you were talking about physiological sort of, like endogenous-like type of manipulations, I don't think we're there yet.
I should say more physiological.
Definitely. No, I would agree with that.
Yeah.
But I just want to point out the caveat that we're still, we still don't have the tool that would give us the ability to, do a manipulation that would really mimic the way the activity is normally happening in the brain.
See, this is a good illustration of taking a perfectly simple story. There's phasic and there's tonic, and just finding all sorts of problems with it and issues, and there's a complete gradient between phasic and tonic, and maybe there's an analog computation within that gradient, blah blah blah. And it seems like if the brain can do something with the tools it has, it does. And I don't. Maybe this is a good time just to segue into the panel discussion topics.
Oh, the panel, right?
The panel. Come on, guys, rein it in. No, but I think that this was a good illustration of the complexities involved in these sorts of things, rather than now all of a sudden deep reinforcement learning looks like a very simple, clean story relative to like what's actually happening in the brain, which is presumably these are all based on or related to or an analog of dopamine related computation in brains. So maybe just kind of to segue into that, into the topics that we discussed in the panel, I don't know if you guys have thoughts about how the panel went, or if anything surprised you, or if it was kind of on par for the deep reinforcement learning kind of discussion.
I don't think I was really surprised. I was hoping that we would go a little bit more and talk about dopamine, I guess, because of the nature of the conference, and that didn't happen too much. But I think that's sort of a reflection of where the field is right now, which is sort of, we're still grappling with the idea of deep learning vis-a-vis brain learning. And we're sort of stuck a little bit in trying to figure out the differences and similarities and how to actually relate that. And I think that most of the people that work with DeepRL, they don't care about dopamine at all. I mean, they either care about, doing better AI, or perhaps care about, maybe relating, AI to behavior. And I think what's really missing is the third part of this triangle, which would be the circuit itself. And I think that part seems to me, and Ali maybe has a different perspective, more removed from the conversation related to DeepRL right now. But that was the part that we were like trying to focus more, I guess, in the conference. But I think the panel sort of reflected where the field is, which is more, thinking about the AI part and perhaps related to the behavior part, maybe more high level part, more high level view, and a bit less, more removed from the circuit part.
There was circuitry talk in the beginnings. And I don't know if it continued throughout. And I tried to keep bringing it back, but to your credit,
you did good, Paul. You did good.
I'm not. Yeah, thanks, man. I was feeling down about it. But to your credit, one of the question within the abstract is, can these new advances in deep reinforcement learning help us understand the role that dopamine plays in learning? So, but you're right, it was not very dopamine focused most of the time.
Yeah, I mean, one thing that surprised me, I think, was Matt Botwinick's self-acclaimed radical idea towards the end that brains are deep learning agents, or something to that extent. Deep learning,
deep reinforcement learning.
That's right. Deep RL systems, which is interesting. I'm not sure if I agree with that, right? Because, I mean, how would you define deep? I mean, maybe these are just loaded words. You can define anything to be deep reinforcement learning, right? But is it true? Is it true that brain is implementing a reinforcement or a deep reinforcement learning algorithm? Are we by this discrediting all the other cognitive aspects of brain function, for example, like the memory systems, right? That are somehow again involved with dopamine, or either it's hippocampal episodic memory related systems, or prefrontal working memory type systems. I mean, these are involved in learning in many kinds of actually tasks that we train human and animals to perform. It's not just a simple reinforcement learning algorithm like a Q-learning, actor-critic, or any of these systems that are involved. So I'm not sure. I mean, yes, maybe if you define
sure?
memory to include what?
You're not sure?
I am not sure if I mean, there's an appeal to this, and I think I blame physicists for this, for coming up with a unifying theory that's wrap everything. I don't know if we can do that about brain or not, because brain, I guess it's true for physics as well, but brains were not designed. They were evolved. And one of the fascinating ideas in neuroscience in general for me is that you have these multiple different systems that were evolved during different times for different purposes. And then they compete with each other for behavioral expression. So if you're learning to solve a two-armed band-aid problem, for example, I mean, if you're designing an algorithm, you can design a reinforcement learning algorithm that would solve this beautifully and perfectly using TD reinforcement learning algorithms. Use just an RPE and action values, etc., to solve this. But when you train a human or train a rat, a monkey to do this, are they just doing it the same way that you do? And I think this goes back maybe to a broader question or affliction of neuroscience these days, that if you can explain an animal's behavior using a computational model, can you there infer from this that brain is using that specific process to solve this problem or not? And I would like to see more conversation about that. I think this was a blessed panel. It was the start of a conversation, but in the coming years, I would like to see more and more conversation about what is it that we can learn that's are we at a point that the divergence between artificial intelligence and neuroscience has gone so far that there's no way, I mean, these are now different, totally different processes, different disciplines, or are there things that can each learn from each other? And I think it was a good start for this conversation.
Okay, so that was very interesting. I want to remark on several things. I won't have time, so I'll just pick a couple. So the first is that, I mean, you were very, very polite when you said, I'm not sure whether the brain is an deep RL system. Look, I don't think Matt believes that the brain is it was obviously sort of, putting it in a sort of an extreme way, provocative. And obviously he works on deep RL system, but clearly we know that the brain does so many different things in so many different ways that it cannot just be one thing. I think the question is whether there is reinforcement learning happening at all in the way that we think about in the brain. And I think that sort of the dopamine story points towards the answer yes. So far, you would have to bet. I would bet yes. How exactly it's implemented, I don't know. Is it the only thing that's happening? No, it's not. But certainly, I think this is, these extreme views, even the people who say them, I don't think they really believe them fully. It's just, a way of starting a conversation and sort of pointing out to a direction which could be useful. And so regarding your last point, I actually think a little bit of the opposite, which is I think that AI and neuroscience are actually coming closer together. And the reason that sort of inspired me, got me excited about this panel, is the fact that in DeepRL, you have something of a very poor version, perhaps, of a circuitry, as compared to all the previous reinforcement learning models. And that's something that I looked at that when I first looked at this idea of DeepRL and said, wow, okay, so we're getting closer in the sense that we might have something to anchor to, because one of the problems that we have is exactly what Elise said, which is you have a model and you try to replicate animals' behavior. And if you're very good at tweaking your model, you'll be able to do it, right? Because that's what models do. They're very general typically. And the more general it is, the better your chances are to tweaking it and then sort of recapitulating a behavior. And then you can say, well, maybe the animal uses that model. And there's no great way, like Ali said, of actually validating that. And for me, the validation that would satisfy me would have to do with a prediction about the circuitry that would be maybe even independent of the animal behavior, which we could then go ahead and sort of probe the brain with an experiment and try to see whether that, whether that's being validated or not, where we get something close to what we thought. And so for me, when I thought about DeepRL and dopamine, I said, all right, now we have a way maybe towards, looking at the circuitry that would be predicted by the model and getting experimental predictions from that and then going to the brain and trying to do an experiment where we could test those predictions. And so my sense is that ultimately, whether the brain does something like DeepRL, for me, I would like to see a validation that involves a prediction about the circuitry that's being proposed by the model that then is being validated by an experiment.
No, I think that's a good idea. So, I mean, basically what I guess you're suggesting is that like Mars level 2 should inform us something about like level 3, perhaps, which is interesting.
Right.
I mean, yeah.
Right.
Level 3
being...
I
never, because if we just stay on the behavioral level, again, you can always have a model that does whatever behavior you might find. And if the behavior is the opposite, you can find a model that does the opposite too. I mean, that's not, when you have something general, I mean, when I started, I was doing motor stuff, motor system, motor cortex, and people had this idea about how motor cortex codes for movement, which is in a sort of like vector way, all the cells sort of vote to which direction the arm moves and so on. And then came the idea about optimal control and how what the motor cortex might be doing is to minimize a cost function that would enable you to move, like an optimal control system, the way an engineer would think about it. And one of the main successes of this idea was to recapitulate all sorts of behaviors that people or animals did, the way they moved, the kinematics of the movement and so on, the way they would overshoot their target or undershoot in different type of tasks. And for each of these papers, and I'm being very flippant and unfair on purpose, they would change the cost function in a way that would allow them to recapitulate that behavior. And again, I'm being unfair, so I'm not being 100% serious, but I want to bring the point that, when you have a general enough theory and you can play with it enough, then recapitulating a particular behavior might not be that strong of a proof. And that is why I would like to sort of mess up the Mars levels and go to the circuitry and get predictions from that to help us, get a stronger proof about whether this model is actually related to what's happening in the brain.
Yeah, fair. I mean, just for me to wrap up maybe, and then I'll shut up soon. I think the way I see it, actually, the dopamine system is a very good example for the MAR 3 levels. And actually, the link between dopamine and reinforcement learning, that at the first level, what I see is that there's a problem to be solved, which is how to maximize the expected sum of future rewards. This is the problem that reinforcement learning tries to solve. And whether or not brains or like natural agents are doing that or not, I'm not quite sure. Economists would tell you that, yes, this is exactly what human brains are designed to do, just maximize future rewards. I'm not quite sure about that. But reinforcement learning proposes this series of algorithms. And, we have the old ones that I've read are like Q-learning, actor-critic, SARSA. etc., etc. And we try to link them to some brain function to find a mechanism that would implement these functions. So this is the part that I'm not sure whether or not we can get a good link between these like algorithms and the way the mechanisms they're implemented in the brain. But like what I see, maybe just to say a sentence or two about how I perceive this conference beyond the panel. There were a few good things and hopes, lights at the end of this tunnel, that we are getting better tools. I mean, we saw a few talks that are introducing new tools, including like Ehud Isakov's like amazing new tools that would help us figure out a few things about better and better about mechanism. Like in the past 2 or 3 years, the dopamine research field has like shifted tremendously because of the advent, I think, of these like fluorescent dopamine sensors from like Lin Tian's lab and others. And then I saw a few glimpses of a good observation leading to theory. One that I particularly am very fond of was Melissa Warden's observation that how dopamine ramps can be linked to cognitive maps or to exploration of this idea of cognitive maps. I think this is what we need more and more in neuroscience, to find the mechanism, to make an observation, and then try to come up with a theory, at least, that links this to some behavioral function. And I saw an example of this in Melissa's talk that made me hopeful that we're making some progress, at least.
Yeah, I love that. And that's exactly sort of the mixing of the levels that I'm very fond of, because that's really, that gives me much more confidence that it's rooted in reality when I see that it's sort of, it bears out in at least 2 different levels. And I think that's what we need to do. And I agree, people are doing that. And of course, we're limited by the tools we have, but even with the tools we have, there's lots more things that we can do.
So what I was hoping for is to give some context to the panel here, and what I expected happened where I got a lot more. Did you guys get everything in that you wanted to talk about?
I think so, yeah. I mean, I have a few other points that are like random points that I wanted to talk about, but I think we can. I mean, one of them maybe I want just to say here because it links to your show, which I'm a big fan of, by the way. And I like to keep it like in audio form, not make it a talk show, because I used to listen during my commutes, and it's been very informative for me.
Oh, good, thanks.
I mean, you had Matt Smith on your show, and I forgot. I think it was episode 85, 86, 90
you forgot?
Something. I forgot. I'm very sorry.
Seriously?
I apologize.
It's all up here, buddy. Yeah. No.
And he was talking about this fascinating idea of slow drift, which I think it's an amazing observation. And I like to link that to dopamine and neuromodulatory systems. Mm-hmm. I think it's a fascinating avenue for future research. And this may link to like more computational or like neural network architectures as well, that what we currently are inspired by in our neural nets are like fast neurotransmission, meaning like excitatory inhibitory connection, right? That's like this unit A, neuron A fires and then it would excite or inhibit neuron B. And this is like a rapid summation in one step. But what neuromodulators do is that they don't necessarily have an excitatory or inhibitory effect. They change the state of a neuron or a network in a way that they would respond differently to future stimuli.
Is this like Roshan Kuhl's work on switching states? Is that kind of thing?
Maybe it goes even back to like some of the ideas that I've seen abandoned now by like Dershowitz and Jeremy Siemens. That's like how like dopamine changes the state of a network. Like different dopamine receptors, when activated, would move you through this like state space to different like basins of the attractor, which I think these were like amazing theoretical ideas that have not been explored. And what Matt talked about in your one of the previous episodes reminded me of that dopamine has this role in like attention, for example. And these slow drifts might have been related to that. And we currently have no way of incorporating these in our computational models in our artificial neural networks. And this might be an avenue to do something which is like, yeah, I see myself at least in the future doing that. I proposed this to the Simon Foundation, but I think it's a fascinating idea, and we'll see more of this in the future, how like these like different neuromodulatory signals, like dopamine is one, acetylcholine, serotonin, norepinephrine, how these not non-neurotransmitters, these neuromodulatory signals change the state of the network. And if there's a connection back to the AI, maybe this is something that we can contribute to their understanding. Maybe this can be one of the wings of that plane.
I was originally going to ask you, and now I'm going to bring it back in, about your thoughts broadly about A, where are we in understanding dopamine and its roles, like broadly, right? But then B, I wanted to get back to this idea of, the idea of neuromodulatory modulations being slow. And traditionally, when you think about computation, you think about binary, you think about spikes or not spikes, ones or zeros, and that's how you compute things. But do you think we're trending toward a multi-temporal scaling story of computation where neuromodulatory effects might be a slow part of that computation, whereas the spiking is just another faster part of the computation, and where we're going to get this eventual hierarchy of kind of scaled temporal computation? That's a lot to think about, I know, and those were two different questions. But if you have thoughts on either of those, I'd love to hear
So
them.
in response to your second question, maybe first question, let's talk about the first question. I think we touched on that a bit. I think we are learning more and more. I mean, as we are graduating from this dopamine RPE theory, which is, again, an elegant account of dopamine function.
I like the graduating term there. Yeah, sure.
I think we are going back to test out some of the older ideas, and we are and as we get like better and better tools for probing dopamine function, we are starting to explore the role of dopamine in different functions and different aspects of behavior, as some people have shown in like a series of talks during this virtual dopamine conference FIDA. Mm-hmm. But in response to your second question, I think that's very true, right? That we need this to like somehow introduce this idea of processing at different time scales. And my pitch to the Simon Foundation for this idea was that, I mean, if you look at behaviors evolve over a very different time scale. Right? Whereas like neurotransmission is super fast. It happens in like milliseconds. But the time scale of behavioral changes are much slower and much different. You have different behaviors that happen and pan out at different time scales. And what could link these two fast processes and slow behavior could be the neuromodulatory systems. And as like Ben was suggesting earlier, for example, like dopamine as like a usual suspect of all neuromodulation. I mean, you have this idea of that, yes, dopamine cells fire and then they dump a bunch of dopamine in different parts of the brain. What happens is that then these different parts will absorb this dopamine, and they have different rates of absorption because there are different mechanisms involved in the absorption. For example, in the striatum subcortical regions, you have transporters that will just suck out dopamine and put it back inside the cells. And the density of these dopamine transporters are different, even in different subregions of the striatum. But then in the cortex, you don't have dopamine transporters. It has to be dopamine has to be metabolized, and that's a much slower process. So dopamine is hanging around for much longer in different, up to like 30 seconds perhaps, right? If you get a simple puff of dopamine, it will like hang out there for much longer compared to like striatum, where it gets sucked up within a second. Right?
You mean like a tonic signal, right? No, I'm just...
let's not get into that. But so, I mean, and the reason I think this is an appealing idea is that if you look at these like different modules of the brain, different parts of the cortex or different parts of the brain that have evolved and have these like local microcircuitry involved, But then for the neuromodulatory systems, there's always one region, one nucleus, or at least mostly one nucleus in the brain that projects to like all over the brain and would broadcast the signal. So that's another appeal of this dopamine as a broadcast signal, that you have dopamine cells sitting in the midbrain and then they just dump dopamine all over the brain. So that to me is something that could change the states of the brain. This, how you process. You get more dopamine, you process things differently. Now the same inputs would mean like a different thing. We had this recent paper, that came out in Science from Adam Kepich's lab, where he was looking at hallucinations or hallucinatory perceptions, HALOPs, I think they call it. And the idea was that sometimes the mouse hears things that are not out there, thinks that they heard something and then they move. And what they show is that how this is related to the dopamine levels in one particular part of the striatum, in the tail of the striatum in the back. So I think, the same cues then would mean different things, or the absence of the cues. These neuromodulatory signals change the state of the network, and how these networks process information, or even same information, would mean different things. So that's my thought.
Yeah. I think that the issue of timescales is actually even worse than that, because even if you just talk about regular neurotransmitters and not neuromodulators, you still have different synapses can have very different timescales. And in fact, if you look at computational models, some of the models, depending on the timescale of the synapse, you can have a very different type of behavior. There's some rate models where if you have synapses that are slower than 20 milliseconds, let's say, then they behave in a completely different way than they would behave synapses that are faster in like 10 milliseconds. Okay,
Mm-hmm.
so even within that timescale, the network itself can be very different, and you can explore that computationally, depending on, the timescale of the synapses by a few milliseconds of change.
Yeah.
And then, of course, you have the neuromodulators, which right now are being modeled either as very fast or very, very slow, because let's remember synaptic plasticity, right? That's sort of the big effect of the neuromodulators like dopamine in these computational models. And that's a very, very slow, a time scale. And so, but of course, within that range of, a few milliseconds versus, minutes, let's say, you still have what Ali talked about, which is how, you can have fluctuations in the way the neurons change its state and the network change its state. And that has been mostly, I think, explored by cellular physiologists, a bit perhaps less by computational or even maybe systems neuroscientists. They're starting doing a little bit now, I think. And so that's adds a new layer of complexity, which is something I like. I like complexity. Maybe that's why I'm studying the brain. And just to piggyback on this idea of complexity, Ali, your idea of the appeal, I understand the appeal of the broadcast signal, where you have the same signal that goes everywhere in the brain, and then different brain areas will process it differently depending on the kinetics of the reuptake of dopamine and depending on their own characteristics of the network. But even that story, I would argue about a complexity, because we know that different dopamine projections can transmit different signals to different areas. And so even then you have this added complexity within the same nucleus, where you have this concentration of dopamine releasing neurons or other nuclei. When you have sort of, let's say, other neuromodulators that's being released, you can still have, different streams of information that's being transmitted to different places as well. So I think that the more we're going to delve in, the more we're going to find that we have this further and further subdivision and further complexity because of this. Again, this is something that Ali said, and there was also alluded in the panel that brains evolved, and they evolved in a way that's sort of disorganized and chaotic and just get things to work. And that, I think, naturally results in a huge amount of complexity.
Is one way to think about this. So recently there's been a lot of success in using dynamical systems theory to basically reduce the dimension of our brain, of the activity in our brain. So our brain has tons of neurons and they're all firing, but you can reduce the dimension so that the neural firing is fairly bound to a lower dimensional manifold, a lower dimensional shape, essentially, and its trajectories along those manifolds that underlie some of the computation. But what you just said is that we may need to then increase the dimension if we're considering how different areas are responding in different ways to the same signal, and how that increases the complexity, the amount of interactions going on. And I don't know if it's worthwhile thinking about it in terms of how that story might map on to the let's make everything low dimensional story.
I mean, it's very appealing to make everything low dimensional, because then we can do we can put it on a graph and we can look at it. If it's 2 or 3 dimensions, that's visual, and we're visual creatures. So I think that's part of the appeal. But again, this idea about dimensionality reduction, we need to remember that dimensionality reduction, all the idea about dimensions basically are reflections of the correlations between neurons, right? How much the activity between neurons is correlated or not. That's all it is. And so if you have a lot of neurons that are highly correlated, even if the pairwise correlations are not big, but the correlations overall are strong, then you will be able to do this dimensionality reduction. If you have a very low correlation, the neurons are independent, everyone is doing their own thing, then you can't reduce the dimensionality. And so I think it's clear that, neurons are connected, so they're going to have some correlations, and the more neurons you record, the more saturation you're going to have on the amount of information, which allows you to reduce the dimensionality more and more, relatively speaking. But of course, that's typically done within a single brain area, maybe even within a single condition. And I think that the more you go into multiple brain areas and receiving multiple inputs, including multiple neuromodulatory inputs, then it's going to be harder and harder to just think about a 2D space that you can nicely plot your graph in different colors and call it a day.
I mean, there is the visualization aspect, but there also is the aspect of figuring out where the saddle points are, figuring out where the attractors are, where you can predict what state the system will be in given its initial state and the condition, right? You will know fairly accurately if you know the surface of the manifold, the shape, the topology, then you know where the activity will end up, right? So there is a computational advantage as well.
I think that's independent of the dimensionality. I mean, you could have attractors, you could have attractors with completely independent neurons as well.
Sure.
So I think that it's easier to look at the attractors in a lower dimensional space.
Yeah, for sure. Ali, anything to add? We've gone off course, but that's fine.
Yeah, I mean, no, I think, I have my own, interests and skepticisms about these, dimensionality reduction thing. I agree with Ben that they're, amazing visualization tools. And I think it's a good descriptive language for, what computation, the circuit or specific circuits, might be doing. But I'm not sure what we would learn beyond that about the, circuit dynamics or circuits underpinning circuits of a certain behavior, right? But I think absolutely it's a good descriptive way of computation.
I guess I was bringing it up just because it seems like a hopeful story of being able to visualize and use these lower dimensional structures to simplify things, and that's against what Ben loves, and he is saying, oh, good, there's more complexity with the neuromodulatory story. He's rubbing his hands together. That's what you can't do on a audio only podcast, to see that someone's rubbing their hands together.
So let's add to the complexity here. Yeah. So Ben alluded to this idea of this is one of the hot topics now in the field, that during a virtual conference meeting, we also saw some traces of it, that there's a disconnect between firing of dopamine cells and the amount of dopamine release. Ben's laughing, but the idea is that...
I love it.
Yeah, it's been a few years now that we know that dopamine cells are not exactly your typical textbook cells, that the amount of neurotransmitter here, dopamine, that they release is not a passive readout of the cell activity or the cell body activity. I mean, in your typical neuroscience textbook, you read that cells fire action potentials, and then proportional to that, they would dump neurotransmitter in their targets. But when was the first study? I forgot. But even if you go back to some of the Schultz's old stuff from 80s, people were suspecting that if you get a brain slice where you cut out all dopamine cells, the cell bodies, and then you have just the processes in the striatum, for example, just cut it dump them in the trash. And you stimulate that, you do an electrical stimulation in the same slice, and you measure dopamine. You would see dopamine release. So there are mechanisms, and then later out in 2012, using optogenetics, Steph Craig's lab and it was Joe Cheer, right? I mean, Roger Kachop. And at the same time, what they discovered is that if optogenetically you stimulate a certain type of interneurons in the striatum, these are interneurons that release acetylcholine, then you would get dopamine released in the striatum independent of firing of dopamine cells. Dopamine cells are, like cell bodies, are dead. You cut them out, throw them in the trash, and you stimulate these certain interneurons, and you would get like dopamine released. So this adds, I think, even more complexity that, like, what is the computational unit of the brain? I mean, is it like firing of or of cells? Or maybe in this special case, it's not that. It's the amount of dopamine that's getting released. Or even maybe further. I mean, maybe we should reconsider about what computational unit of the brain is and think about like receptor activation. Because in the end, if dopamine cells fire and there's no receptor to listen to them, have they made any sound?
What about water? Because the brain's mostly water. Should we consider that the computational?
It's salty water, right? If it was water, we wouldn't be alive, right? It's salty water, and it's actually cells that are trying to get a shelter from that salty water. And the gradient there, I think, is the heart of computation there. But yeah, I think it's the receptors, yeah. So, yeah, I mean, this adds to the complexity that dopamine cells it's not enough just to record from dopamine cells and infer their function. And, like, during this video conference, we saw, like, amazing results from Zaid Khalik's lab. Paul Kramer showed some amazing stuff, and Chang Lu from Pascal Kaiser's lab showed, like, another set of amazing results showing how the interaction between cholinergics and dopamine axon terminals in the striatum would cause like dopamine release. And we don't know yet, but we are trying to, like, multiple groups now are trying to understand what are the functional meaning of this now added computation. And I have my own speculations. I mean, it has something to do with like saving. I mean, I'm just babbling, saving like metabolic energy, because I mean, one thought that I had was that, like, dopamine, unlike other cells, I mean, the branching of dopamine is like maybe 2 or 3-fold greater than your typical cells. So changing even the membrane potential would be like very metabolically costly. There have been like computational studies out there that shows that the energy in terms of ATP required to bring dopamine like axon terminal potentials back to a resting potential is like much greater than your typical cells. And it like exponentially increases with the number of branching. And one reason is that maybe brain, I mean, I love this idea of evolution of systems sitting on top of systems. Maybe at some point they were like easier or more simplistic mechanisms or, agents lurking around the earth, and they just needed dopamine to do this reward prediction error thing, or like 2 or 3 more things. But then as things became more and more complicated, brain learned to design this idea of let's do a local release of dopamine in this special circuit, because this circuit maybe cares only about motivation. So you don't need to broadcast dopamine all the way from midbrain to the entire brain and then use all this energy to bring back all these terminals down to the resting membrane. You design or you evolve this local mechanism for dopamine release. I think this is a topic that maybe like 10 people care about in the entire universe. So
lots of people care about it. I actually think that, both things are probably happening, because even when we record from dopamine cells, we see that not all dopamine cells do the same thing. And so you can have that sort of complexity, and then you can add to that the local circuit, sort of independent modulation, that Ali talked about, to have another layer of complexity that even those different signals can be even then further sort of distributed, further modulated, in separate ways. And that gives you a lot of control, quote unquote, that's kind of a loaded word, about being able to do lots of different things with this type of neuromodulator. So I think that's a good thing. Again, I like it.
Yeah, and going back to maybe your first question, this is that there are aspects of, like, there are functions of the dopamine that are, like, essential to life, like learning from rewards and aversions, right? So the entire brain perhaps needs to be informed about this function, that something good happens, right?
That's what Matt said. It's the whole brain is a deep RL system. There you go.
The whole brain has to be informed. The whole brain is involved, perhaps, in deep RL, but it's not. But yeah, and there's work from, like, functional MRI stuff from, like, Carl Dijsselrath and others' labs looking at reward signals or reward prediction signals across the brain, and you see that, like, brain is involved, right? So it makes sense to have a broadcast signal that would just send out these, like, alarms, or, like, something good happened, something bad happened, right? If you're the visual cortex, perhaps you still need this. If you're motor cortex, you need this. But then there are, like, very specialized functions of dopamine that perhaps not every single circuit in the brain needs to care about, right? How to hold a thought in your memory. Perhaps prefrontal cortex wants to know that. Your visual cortex is, I don't care about that. Let me do my job, right? So these maybe mechanisms evolve. And I agree with Ben that, yes, it's not that, like, there might be heterogeneity, and there is, like, at least we know through his work and others that there's heterogeneity in responses. But on top of that, I want to propose that there might be a multitude of local regulation mechanisms for dopamine release that serve these specific functions that we are yet to discover.
Yeah, and that's, I think that's really well supported, because even though there is heterogeneity in the activity of different cells, when it comes to the big sort of salient behavioral continuities like reward or aversion, at least reward, you can see that most dopamine cells really respond to it, regardless of what they do in different kind of epochs. So certainly, that's something that dopamine neurons seems to be specialized towards, even if they do other things as well that are more sort of segregated or heterogeneous.
All right, guys. Well, this is what it was going to be at the beginning was a sort of cursory, introductory, reflective precursor to the dopamine panel, and what it turned into, which is what I thought it would do, what it turned into was a full-blown discussion that I think has turned into a real nice complement because it was focused a lot on the actual dopamine, on neural systems and ideas surrounding those things. So then the next little episode with the panel will focus more on that computational AI and behavior side. So thanks again, and thanks for having me again, and this has been great.
Thank you for having us.
Yeah.
Brain Inspired is a production of me and you. I don't do advertisements. You can support the show through Patreon for a trifling amount and get access to the full versions of all the episodes, plus bonus episodes that focus more on the cultural side but still have science. Go to braininspired.co and find the red Patreon button there. To get in touch with me, email paul@braininspired.co. The music you hear is by The New Year. Find them at thenewyear.net. Thank you for your support. See you next time.
The stair